In this video, we will discuss about different functions, different types of T-effector cells, and how this T-effector cell scans the normal cells or the tumor cells within the host and identify whether that cell has some kind of foreign antigen present in it, or it is not present within the cells that T-effector cells is scanning. So let us first understand how these T-effector cells function. Once it identify that the cells has this antigen component that does not belongs to the host, or it is a non-self antigen. So the first type of function that is commonly known as is that these immune cells will detect the virus infected cells or the foreign antigen on the cells and will trigger the apoptosis that can be seen here. In this case, these T cells has these CD8 plus receptors, and it also has the information about the non-self epitope. With the help that information that is received to these T cells through this with the help through dendritic cells, and with the help of this major histocompatibility complex. So now this CD8 plus T cells has the non-self antigen information, and whenever it detects any non-self antigen, it triggers the apoptosis of those cells. This is how one of the way by which these CD8 plus T cells function, that can be seen here on the left hand side. The other way this T cell function is also categorized as this Th1 cells. So in this case, what happens whenever it detect non-self antigen on the cells that it is scanning, it will trigger macrophages within the cells, so that those intracellular irregularities such as intracellular bacteria will be killed off by the macrophages that is activated within these cells through this Th1 cells that can be seen here. This macrophage will kill the bacteria within the or irregularities within this non-self epitope containing cells. The third way that it can work in this case, one more possibility is that these T cells will also activate the B cells to generate the antibodies against this kind of irregularities. So another kind of activation that is done by the CD4 T cells that has this MHC that has this non-self epitope information. With the help of this MHC major histocompatibility complex interaction with dendritic cells. So in this case, this Th2 cells, what it does is it will activate the B cells to generate antibodies against this non-self epitope that is been detected by this Th2 cell. So we can see here in the bottom figure, the B lymphoblast will occur because of this Th2 cells has identified a non-self epitope containing cells during its scanning process. So the major difference between these functions, that is cytotoxicity killer T cells and Th1 and Th2 T cells, is the receptors. Whenever the CD8 plus receptors detect, it will trigger apoptosis, or whenever these CD4 plus receptors detect, it will either trigger macrophage, or it will trigger B cells to generate antibodies against the non-self epitope. Now let us quickly understand how this scanning process works. So the example shown here is about the CD8 plus cells. So CD8 plus cells interact with the cell surface proteins through this LFA1 and ICAM, and this is a non-specific adhesion that will occurs. And because of this adhesion, now the cell will starts to roll and check whole cell surface area to find out if there is any non-self epitope is present or not. And if there is no antigen specific no non-self antigen is been found on the cells, it will disengage itself from the cells and attach to its other and scan the other potential cells in which the non-self epitope could be present. So and whenever it finds a non-self epitope, it will trigger its function based on the functions that we have already, based on the types uh, of T effector response that we have already discussed. And uh, after this T cells release or perform its function, it will disengage itself from those affected cells and starts to rescan another potential target. So this is how this T effector cells function, and it generates this is how T effector cell functions, and it scans through the different potential target. If you need to understand more about these T effector cells and its scanning process, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.